back with your next class. In today's class, we are going to discuss a very important part of transport in plant chapter, that is long distance transport. All this while, we have learned about various processes like diffusion, osmosis, plasmolysis, etc. These were all the transport where materials move from cell to cell, isn't it? But then in tall trees, in the plants, the water or the food, right, that has to be translocated throughout the plant body. And if the plant is a very tall tree, it might have a height of say 100 meters. So in that case, the materials have to be transported to a very long distance then, right? So any kind of long distance transport in the plant body would be occurring by the help of the major conducting tissues, xylem and phloem. Alright, the xylem and phloem will carry the materials to long distances, not only through long distance, rather also the materials that will be carried will be in bulk or in mass. Okay, so this flow of materials or substances throughout the plant body via xylem or phloem in mass or in bulk will be known as mass flow or bulk flow as well. Alright, and the movement of materials through the help of xylem and phloem is also known as translocation. So we'll be discussing here about translocation, how translocation occurs through xylem and through phloem separately. Let us start with xylem. How transport of materials would occur by the help of xylem. Alright, now see, transport via xylem mainly would occur by three stages. One, you have to understand how water moves from soil to the root hair. Next, you have to understand how water from the root hair moves up till the root xylem. And then how from root xylem water would be transported up to leaf xylem. Alright, so these are the three phases in which water would move via xylem. Okay, so after all, xylem being present throughout the plant body, do remember the water or minerals that is transported via xylem will always be moving unidirectional. Okay, xylem, it will conduct materials unidirectionally, unlike phloem. Phloem will move the materials bidirectionally, sometimes from leaf towards root or root towards leaf. Alright, so that's a different issue we'll be discussing in another class, but today we'll mainly concentrate in the transport that occurs via xylem. Alright, so now see here, see, whenever xylem has to transport the materials, first of all, water from the soil will be absorbed by the root hair, isn't it? I'm discussing the first one now. How the water from the soil reaches the root hair. Alright. So suppose this is root hair and this being soil. Okay. The surrounding soil. Now concentration of solute is going to be always less in the soil in comparison to root hair. You might be thinking root hair is so minute, it's microscopic. How is root hair going to have more of solute than the soil? It's something not very convincing, right? Now, listen to this. Whenever you measure concentration, it has to be according to the available space. Like, suppose in a bucket of water and in a small glass of water, I add 10 tablespoons of salt. Okay, in both. Where do you think salinity will be more? Where will the water be more saline? Of course, in the glass of water, isn't it? Why? Because it will have less amount of space in comparison to the solute that is being mixed, right? So accordingly, now if you apply this here, you can easily understand how the solute concentration in this microscopic root hair will be much higher in comparison to the surrounding soil. So now that you know that solute concentration in the root hair will be higher in comparison to the soil. So in that case, don't you remember by as osmosis's definition that water always moves from a region of lower solute to higher solute via semi-permeable membrane. That will be the plasma membrane. Alright. So soil to root hair, the water would be absorbed simply by endoosmosis. Alright. Now, once the water has been successfully taken up by the root hair, now if you try to recall anatomy chapter, you will remember that root hair up till the xylem, there will be a lot of layers, okay, cell layers that the water has to surpass. See, root hair is a part of the epidermis, then comes general cortex as hypodermis is absent in roots, then comes endodermis, then pericycle, and finally comes the xylem, right, the vascular bundles. So all these layers from root hair up till xylem, there are so many layers to be surpassed. Now, how will water surpass all these layers? That is what we are going to discuss in the second. See, root hair up till root xylem, how the water would move. Let's see that. Let us first make a diagram. Suppose this is the root hair and this is the epidermal cell followed by general cortex. Then comes endodermis, then comes the pericycle, and finally we have the xylem. Alright. The water from the root hair has to successfully reach up till the xylem, right? This is epidermis, general cortex, this is endodermis, this is pericycle, right? This being the cell wall, and you know very well that in the plant body, each of the living cell will remain interconnected by the help of plasmodesmata, right? So this is the plasmodesmata that runs through pits, the sap vacuole, and of course the nucleus. Now, water has been absorbed by the root hair. Now the water, it has to move through this entire layer, okay? Now see, water, when it wants to move through these layers, it would of course want to take a route that will provide least resistance, right? So water has two pathways that it might choose from, it might prefer to go from. Like, one is the water may move through the cell wall because cell wall is freely permeable after all. Another route is it may pass through the cytoplasm via plasmodesmata, it might move. But if the water chooses to go through the cytoplasm, it will have to pass through the plasma membrane, which is not very welcoming to the water after all, because the plasma membrane has a hydrophobic core, right? So in that case, water would be mostly moving through the freely permeable cell wall, okay, through the interconnected layers of cell wall, okay, through general cortex and epidermis. It will move mostly, yeah, a little bit of water of course will pass through cytoplasm as well, but then mostly water will pass through interconnected cell wall as it will provide least resistance. Alright. But then, if you remember about the cell wall of the endodermis, the cell wall is going to possess a suberin deposition that's called Casparian strip. And suberized cell wall are impermeable in nature. They won't allow anything to pass through. So now that the water reaches the endodermis, does it have any other option to pass through the cell wall anymore? No, right? So now the water will have to move into the cell cytoplasm. Alright. And from endodermis pericycle, it will have to move through the cytoplasm only. No more cell wall's route can be taken. Alright. So now that the water will have to pass through the inside of the cell, Again, there can be two routes that can be possible. One is water may ignore the sap vacuole and pass through the plasmodesmata through peripheral cytoplasm and reach the xylem. Or else, the water may pass into the cytoplasm, move to the sap vacuole and then move from one vacuole to another and reach the xylem. Okay, that means it may take up the route of the vacuole through vacuole to vacuole, or else it might take the peripheral route ignoring the vacuole. Why it would can you tell me which one will be now easier for the water to move through? Will it be the vacuolated pathway or the non-vacuolated one? Which one will provide least resistance? Of course, the non-vacuolated one. Why? Because if the water passes through vacuole to vacuole, in that case, the water is having to pass through two membranes. One is the plasma membrane, another is the tonoplast. Tonoplast is also the same, just like the plasma membrane, it is semi-permeable. So, it is hydrophobic in its core. Okay. So, water, if it passes through the vacuolated system, in that case, it will be more resistant in comparison to the non-vacuolated one, as in non-vacuolated one, the water is having to pass through only one hydrophobic membrane, that is the plasma membrane. Understood this? Fine. So, from epidermis and general cortex, we see that most of the water is now passing through the cell wall, interconnected cell wall, and interconnected cell wall is called apoplast, which I have told you in the cell chapter itself, right? So, while the water is passing through the cell wall, the freely permeable membrane, that would be referred as apoplast pathway. And when the water chooses to pass through the inner part of the cells, that is through the plasmodesmata, that is, it is passing through interconnected cytoplasm, right? So, interconnected cytoplasm would be referred as symplasm, if you remember. So, we will call this much part where the water moves through symplasm to be symplast pathway. And symplast pathway, as you have seen, can be of two types again, vacuolated and non-vacuolated. So, which pathway will provide maximum resistance to the water? Vacuolated symplast pathway. And which one will provide least resistance? Of course, the apoplast pathway. Is that clear? So, root here to root xylem, the water would be moving through apoplast and symplast pathway. Alright, understood this? Fine. Now, finally, once the water has successfully been uploaded to the root xylem, from the root xylem, the water has to now move upwards and reach the leaf. Okay, it has to reach the leaf xylem. And this upward movement of water is known as ascent of sap. And ascent of sap, let us discuss next. So, regarding ascent of sap, the upward movement of water and minerals through xylem, once the water has been uploaded to the root xylem, how it reaches up till the leaf, that's what is called ascent of sap. And ascent of sap can be discussed by two theories. One is root pressure theory. Another is transpirational pull theory. Let us first discuss root pressure theory. root pressure theory was proposed by Priestley and was coined by Burgestein. Okay. Now regarding root pressure theory, first thing that you should know is that, see, in the xylem, the conducting elements are the tracheids and the vessels, right? Tracheids and vessels, both are very much narrow, right? They are very narrow in diameter. So as soon as water enters the tracheids and the vessels of the xylem, the water is enclosed in a very, or it has been entrapped in a very enclosed space, isn't it? Whenever water is entrapped in a very enclosed space, a positive hydrostatic pressure will be developed in the space where water has entered. Okay, so in the tracheids and vessels, directly a positive hydrostatic pressure will be built up. Okay, now due to this pressure, the water would start moving upwards through the tracheids and vessels automatically. And this upward movement of water is going to happen because of the pressure, the positive pressure built up in the tracheids and vessels, which is known as root pressure. Understood this? Fine. So water gets uploaded to the narrow tracheids and vessels. As they are very narrow, that's why a positive hydrostatic pressure will automatically build up in the tracheids and vessel as water is uploaded there, due to which water would start moving upwards, and this is what would be termed as root pressure. This positive pressure built up is called root pressure. Alright. But then root pressure is of not very high magnitude. It has a magnitude of hardly 1 to 2 atmospheric pressure, which can take the water upwards only say around 10 to 15 or say 15 to 20 meters, not more than that. It can take the water up to a height of 15 to 20 meters, not more than that. Alright, understood this? That's why root pressure is going to allow ascent of sap, that means water will reach from the root up till the leaf by the help of root pressure only in short height plants, say herbaceous plants. In the tallest of trees, root pressure would only be providing a gentle push to the overall upward movement of water. It will not be allowing the water to reach the leaf. The root pressure in tall trees won't be able to allow ascent of sap. Okay, it will just be providing a gentle push to the upward movement of water. Okay, so the question is, how do in the tall trees then the water would reach the leaf? That would be happening by transpirational pull. Understood this? Fine. Now, even if root pressure is not going to allow ascent of sap in the tallest of trees, root pressure is associated with a very important phenomena that takes place in the herbaceous plants known as guttation. So let's discuss a little bit about guttation here. See, what is guttation? The loss of water from the plant body in the form of water droplet is called guttation. Okay, and guttation occurs due to root pressure. Okay, how? For guttation to occur, the leaves must possess a special type of anatomy. Okay, guttation is loss of water from the plant body in the form of water droplet, and the droplets will mainly arise at the tip of the leaf or in the margin, especially at the vein endings. Okay, in these vein endings, the water would be released. Now, why is this water being released at these sites? That's because if you observe this location or you zoom in, you will find that in the epidermis of such leaves, a gap is going to be present, a pore will be present. This pore is known as hydathod. Just below the hydathod would remain a bunch of loosely packed cells, parenchyma cells called epithem, followed by the xylem directly. Okay, so in the early morning hours or at night, when the root pressure is going to be at its peak, So at that time, water would directly rise up the xylem. Water would move upwards due to root pressure, right? So as the water moves up in the xylem, the water is directly going to reach the intercellular space of the epithelium, and finally it is going to ooze out through the hydatoid. Alright? You might be thinking that in the epidermis a minute pore, so it might be similar to stomata. But no, it is not going to be similar to stomata because stomatal pores are always surrounded by girth cells. Here the hydatoids won't be having as such any controlling mechanism of opening and closing, or they would not be surrounded by girth cells. That's how they are different. Okay? So only by the help of such type of anatomy where hydatoid is followed by epithelium, epithelium by a xylem, and when the root pressure is at its peak, means only at night or in the early morning hours, when transpirational pull is almost nil, it is only at that time that guttation will take place. Okay, that means due to root pressure, water will rise up the xylem, it will go through the intercellular space of epithelium and will ooze out through the hydatoid. Alright, understood this? Now, this phenomena, guttation, it might be kind of similar to transpiration because both are helping the plant to lose excess water, right? But they have certain major differences. Okay, both the process, guttation and transpiration, have some major differences. The differences are, first of all, guttation is water lost in the form of water droplet, transpiration is water lost in the form of water vapor. Second is that guttation is occurring through hydatoid, whereas in case of transpiration, of course, transpiration occurs through stomata. And finally, guttation, it would be losing water. In guttation, water will be lost not in the pure state, rather the water that was directly present in the xylem. And the xylem contains not only pure water, rather water plus minerals. So, the gutted out water will not be pure water, it will be water plus minerals. But in transpiration, the water would be lost in the form of vapor, and of course then it will be pure water. So these are the basic important differences between transpiration and guttation. I hope all of you are clear. So this being said regarding root pressure theory, next let us discuss transpirational pull theory. Okay. Now regarding transpirational pull theory, first of all, transpirational pull theory was proposed by Dixon and Joly, and it explains how the transpirational pull will be created in the plant body, and which is a very high magnitude negative pressure which will allow the water transport via xylem in the tallest of trees as well. Okay, like root pressure would account for upward movement of water up till only 15 to 20 meters. But here transpirational pull will be so high in magnitude, it can carry the water up till 130 meters as well. Okay, so let's see how transpirational pull will be created. Say, this is a leaf, and in the daytime when the stomata is open and sunlight is directly incident on the leaf surface, continuously the mesophyll cells of the leaves would become dehydrated, or they would start losing water. How? The mesophyll cells, they would lose water. They would first, the water from the mesophyll cells would vaporize, and this vapor would accumulate in the substomatal cavity, a cavity that remains just below the stomatal pore. Okay? As the vapor accumulates in the substomatal cavity, obviously the vapor concentration in the substomatal cavity will be higher in comparison to the atmosphere, right? And in that case, the vapor would directly diffuse into the atmosphere through the stomata. Okay? Higher concentration to lower concentration. That's what is diffusion, right? So water vapor will be lost from the stomata continuously, right, due to diffusion. And this is what is called transpiration. Plant is losing water in the form of vapor. That's what is transpiration. So continuously when the mesophyll cells will be losing pure water from them due to transpiration, don't you think the mesophyll cells would become more hypertonic? Why? Because as water is being lost, solute concentration in the mesophyll cells would increase. And as solute concentration will increase in the mesophyll cells, in the veins and vein beds, what are present? The xylem, isn't it? Vascular bundle. So the mesophyll cells will continuously take water from the nearby xylem to compensate for that water loss. Because you know, if you remember, water always moves from a region of lower solute to higher solute. So mesophyll cells are having higher solute now. So of course they will draw water from the nearby xylem. Alright. So the xylem in the leaf, that's why, as it is giving its water to the mesophyll cells, the leaf xylem is becoming lower in its water potential, right? Water from the leaf xylem is moving to the mesophyll cells. So the leaf xylem of course has or will be maintained at lower water potential, right? Whereas in the root xylem, which is continuously absorbing water, here water potential will be maintained at high, right? Water from the leaf is being missed or being lost in the form of vapor continuously throughout the day due to transpiration. Due to that, the mesophyll cells are becoming or having higher solute concentration, and that's why they are drawing water from the leaf xylem. So leaf xylem will be maintained at lower water potential, whereas root xylem, which is continuously absorbing water, will be maintained at higher water potential. So water potential gradient has been created throughout the plant body. And you know very well that water always moves from a region of higher water potential to lower water potential. So this water potential gradient itself will create a pull, due to which the water from the root will rise up and reach the leaf. And this pull is going to be called as transpirational pull, because it is after all being created due to transpiration. Understood this? So this is what is called transpirational pull theory. I hope all of you are clear. Fine. Now, next we are going to discuss another very important mechanism, that is how the transpiration, when the transpiration occurs, we know that transpiration occurs through stomata, and stomata is open during the day and it closes at night, right? So there has to be some mechanism that controls stomatal opening and closing, right? You would say that stomatal opening and closing occurs due to the GERD cells. The GERD cells, when they are filled up with water or when the GERD cells are turgid, stomata is open, and when the GERD cells loses water, they become flaccid. The stomata closes. That's what has been discussed with you in the anatomy chapter. But in today's class or in this chapter, you will learn an entire mechanism of how the GERD cells will get turgid in the day and they will become flaccid at night. So let us discuss the mechanism of stomatal opening and closing. Alright, it is explained through a theory that's called ion exchange pump theory, that was proposed by two Japanese scientists, Imamura and Fujino, but was elaborated by Levitt. Okay, so let's see what will happen or what is the mechanism behind stomatal opening and closing. First, let us discuss stomatal opening. That is during the daytime. See, You know very well stomatal opening and closing is maintained by the Goethe cells, and Goethe cells always possess chloroplasts so that they photosynthesize. So during the daytime, the Goethe cells are undergoing photosynthesis. All these changes are happening in the Goethe cell. Okay. So Goethe cell is undergoing photosynthesis during the daytime, and the product of photosynthesis is sugar, right? That sugar will be converted to starch. Alright. Now, the starch that has been formed in the Goethe cell will be eventually converted to malic acid by the enzyme peptase. Full form is phosphoenol pyruvate carboxylase enzyme. Malic acid, being a weak acid, will dissociate immediately into malate anion and H plus ion. Okay. Now see what will happen. The H plus from the Goethe cell would start moving outside, and it will start moving into the nearby cells. And what is the nearby cells? What are the cells that surrounds the Goethe cells? The subsidiary cells, isn't it? Subsidiary cells, or you can say epidermal cells as well. Subsidiary cells are specialized epidermal cells after all. So say they are moving to the epidermal cells. So as H plus is moving into the epidermal cells, an ionic imbalance will be created, which has to be balanced now, right? To balance it out, potassium ion and a little bit of chloride ions from the subsidiary cells or epidermal cells will start moving into the Goethe cells. So ion exchange has been created. H plus from the Goethe cell is moving to epidermal cells, and K plus from the epidermal cell is moving to the Goethe cells. This is called ion exchange pump, which has been set up between Goethe cell and subsidiary cells. Alright. So now that K plus and malate anion is going to accumulate in the Goethe cell, finally K plus and malate anion together will associate and form potassium malate, which is highly soluble. Okay, it is highly soluble. Because of that, potassium malate will dissolve in the Goethe cells. Okay, and due to that, Goethe cells solute concentration will increase. And you know very well, whenever solute concentration will increase in a cell, it will start drawing water from the nearby cells. So water from nearby cells starts to enter the Goethe cells. Because of that, the Goethe cells will become turgid, of course, isn't it? Water is entering the Goethe cells, so they would become turgid. And as they become turgid, what do you think will happen? Stomata will open. Alright? So this is the mechanism due to which stomata will open during the daytime when photosynthesis is occurring. Now, what will happen at night that the stomata will close? So, stomatal closing. See, at night, the carbon dioxide would start entering inside the stomata as usual. But because sunlight is no more there, that carbon dioxide won't be taken up for photosynthesis, right? Carbon dioxide would be taken up as the substrate for photosynthesis, right? That carbon dioxide itself will be converted into sugar. So now that sunlight is no more present, photosynthesis is not occurring. Stomata being open, carbon dioxide as usual will enter. But then it won't be used up for photosynthesis anymore. So this free carbon dioxide will then combine with water, forming carbonic acid. Carbonic acid will make the entire environment inside the gourd cells to be acidic. So in acidic medium, a plant hormone called abscisic acid will be activated. As soon as ABA or abscisic acid is activated, the membrane permeability of the gourd cell will be reversed, so that whatever was entering the gourd cells will now start exiting it, and whatever was leaving it, it will start entering inside. So it will simply be reversed. So what was going out from the gourd cells? H plus, isn't it? So H plus instead will start entering the gourd cells, and K plus will start moving into the epidermal cells. Alright. So now in the gourd cells, now that H plus is back, H plus will combine with malate anion, together forming malic acid again. Malic acid will be formed again, and malic acid will be converted back to starch. And starch being insoluble in water, as soon as starch is formed, the solute concentration in the gourd cells will decrease. Due to that, whatever water had run into the gourd cells, they will now start moving out of the gourd cells as the solute concentration has decreased. So as the water runs out of the gourd cells, what will happen? So in that case, the gourd cells will become flaccid, and as they become flaccid, the stomata closes. Alright, so this is the mechanism how stomata will close at night. Alright, so this is a very important theory that's called ion exchange pump theory or potassium pump theory. I hope all of you are clear about today's class. So that's all about today's class. In the next class, we'll discuss about mineral uptake or else. So in the next class, we'll discuss about mineral uptake and phloem transport.